I'm now going to introduce Mark Travis. He's the lead of a very interesting branch we've called immune tolerance, where we've tried to bring together different entities that are working on similar concepts. Part of the branch is evolving, part of the branch is established, so drawing together autoimmunity, allergy, placental immunology, etc. They're all basically working on aberrant responses to harmless antigens. So we felt that bringing them together would create innovation at the interface. Mark is a professor of immunology, and in addition to the Lydia Becker Institute, he's also a member of the Wellcome Trust Centre for Cell Matrix Research. So over to you, Mark.
Thanks a lot, Tracy. So similar to the previous talk, as Tracy was introducing, immune tolerance is quite a broad area. So what I'm going to try and do today is give a brief introduction to immune tolerance and then go through a number of our areas of strength and give some specific examples of projects that are being carried out in this area. But my first role as the lead for immune tolerance branch was to actually make sure that I understood what immune tolerance was, because this is a term that is kind of used a lot in immunology, and even sat down with colleagues, we were discussing the subtleties of exactly what this meant. So I went to a textbook, as you do, to say, what is immune tolerance? And essentially, it's quite simple. It's the state of unresponsiveness of the immune system to substances or tissues that have the potential to induce an immune response. So kind of broken down, that's, how we don't make immune responses against things we don't want to make it against. So what are these things that we don't want to make an immune response against and we need to be tolerant to? So we need to be tolerant to our self-antigens and the microbiota that lines a lot of our barrier surfaces, as John has already introduced. We need to be tolerant towards environmental antigens, such as food, pollen, animal dander. If you're pregnant, you need to be tolerant against the fetus, which essentially has half of its genetic makeup foreign to the pregnant mother. And if you undergo organ transplantation, you need to be tolerant to that new organ. And when things go wrong in this immune tolerance process, bad things happen. So if we're not tolerant against self-antigens, we'll develop a variety of autoimmune diseases. If we're not tolerant against our microbiota in the intestine, we'll develop inflammatory bowel disease. If we react against environmental antigens, we'll develop a variety of allergic diseases. If we react against the fetus, this can cause miscarriage or preeclampsia. And obviously, if we react against our organs that have been transplanted, then we can reject these organs. So the aim of the theme is to try and understand how immune tolerance occurs and then how this goes wrong in certain disease situations, and this can help to potentially identify new treatment strategies for inflammatory disease. So there's two main ways in which our immune tolerance is generated. So the first way is termed central tolerance, and this is essentially during the development of B and T lymphocytes in the bone marrow and thymus respectively. There's processes where self-reactive lymphocytes are deleted. I'm not going to go into the nitty-gritty of that. The other way, or the other system, is termed peripheral tolerance. This is essentially once these immune lymphocytes have entered the periphery, the processes, the molecules, the mechanisms by which potentially self-reactive or harmful or lymphocytes that can react against antigens we don't want them to react against, how this is prevented. So the main kind of strength in Manchester is generally in peripheral tolerance, and most of the examples that I'm going to talk about today are generally to do with tolerance in the periphery. So again, just to put into context where immune tolerance fits, there's 30 PIs who are associated with this branch, and there's great synergy with other branches. So this is just depicted here. Over half of the PIs in immune tolerance also have great interest in barrier immunology, and again this makes sense because the barrier, as John's already introduced, barriers are areas of the body that are are insulted by antigens, whether it be the microbiota or food antigens a lot, so this makes sense. Also cellular immunology, we are interested in the cells and the processes that induce tolerance, but then also in many of the other branches, sort of, again, depicting the broad sense of how immune tolerance is important in a number of different themes. So within the immune tolerance theme, there's two major kind of subsets within this branch. So this is the study of the genetic predisposition to inflammatory disease. So we've got some amazing studies with really fantastic cohorts in Manchester, and I'm going to give some examples of those. And then also the understanding of the immunological cellular and molecular mechanisms that promote tolerance. So I'll start with a number of examples of some of the work that's been done looking at the genetic predisposition to inflammatory disease. So in the Centre for Musculoskeletal Research, so Sebastian Viat, Ben Mulhern, Jane Wormington and Anne Barton, as part of the Arthritis Research UK Centre for Genetics and Genomics, are interested in identifying genetic predictors of rheumatoid arthritis risk and severity. So what Sebastian has done, they've been studying a particular molecule, or have identified a particular molecule, so HLA-DRB1. So it's previously been shown that certain amino acids in position 11, 71 or 74 of this molecule could predispose to rheumatoid arthritis risk. But then what Sebastian and others have now shown, that a particular valine residue at position 11 of HLA-DRB1 really increased the risk, the severity of disease. So this is just looking at radiographic outcome as a measure of severity. So just this one amino acid in this one particular molecule increased the severity of disease. So what they're now doing, and Sebastian has a poster on this, poster number 14, they're trying to identify immune cell subsets responsible for variable responses to therapy, to hopefully then be able to link all this together to understand how changes in particular amino acids in this antigen-presenting HLA-DR molecule can alter our risk and severity of developing rheumatoid arthritis. Another great cohort has been put together in University of Hospital South Manchester. So this is the Manchester Asthma and Allergy Study, or MASS. So this is a study that's been going on for over 20 years. And essentially what's been done is that people in Wythenshawe they recruited pregnant mothers and their partners, and then subsequently the children that were born. And then they've basically been following these up since over 20 years, with the idea of determining important genetic, cellular, and molecular markers associated with various allergic airway diseases. Because obviously you've got the genetics of the parents, and you've got the genetics of the children, and then you've also got information as they grow up in terms of the environment, environmental factors that they've been exposed to. And recently there's been big international collaborative GWAS studies that have led to a number of high-impact outputs, and I'll just highlight one of these. So in Manchester, this has been led by John Curtin and Angela Simpson, as I say, in collaboration with many international groups. But this was a massive GWAS study identifying risk loci associated with development of asthma. So this particular study identified 17 asthma loci, 5 of which were novel, and these novel ones co-localized with immune cell markers. And this was recently published in Genetics. They've also done very similar GWAS studies looking for risk in allergic rhinitis. So this kind of links back to what John was saying. We've got this great strength in the genetics, but then we've also got man arts, and we've got great strength in trying to understand the immunology of this as well. And some rarer diseases, so just to point out the work being done by James Lilliker, Janine Lamb, Hector Chinoy on idiopathic inflammatory myopathies. So essentially these are a heterogeneous set of disorders which all result in inflammation and autoimmune reactions against proximal muscles. So again, these guys are interested in the genetics behind this, so they've been carrying out the first large-scale GWAS studies to identify genetic risk factors for different forms of these myopathies, but then also to look into the mechanisms of this. So they've been identifying autoantibodies as biomarkers of disease and potential comorbidities. And then finally, just to point out the work of Tracy Briggs with Ian Bruce, and what they're interested in is these so-called interferonopathies, which is basically the study of single gene disorders that are associated with excessive production of type 1 interferon. So Tracy has a poster on this, so poster 18, so I'm going to talk to her more about this. They're also, they're interested in the genes that are driving these diseases, but also their genetic predisposition to lupus and potential smart treatments, so identifying things like lupus patients who have very high interferon and trying to understand whether you can have smart therapies and identify individuals based on their genetics that would respond better to certain therapies. So that's the genetics. So what about work being carried out on the actual cellular and molecular mechanisms promoting immune tolerance? So as Gloria mentioned, in Manchester there's a great strength in circadian rhythm biology. So essentially circadian rhythm is our body clock. So we have 24-hour rhythm in many of or most of the cells of our body, including immune cells. So most of our immune cells have this intrinsic molecular clock that induces rhythmicity in the production of cytokines and other molecules. And what work by a number of people in Manchester, so Julie Gibbs, Andrew Loudon, David Ray and Hannah Durrington, and others have shown that the time of day can influence inflammatory responses. So this is just work to highlight some early work from Julie Gibbs when she was working with Andrew Loudon. So if you take a mouse and induce inflammation by injecting LPS, if you do this at different times of the day, so essentially CT0 is subjective day, CT12 is subjective night, specific sets of cytokines and chemokines are very much upregulated if you do these injections at night versus in the day. So, and they've gone on in this particular study to show that macrophages are very important in this process. And then very recently, Hannah Durrington, a clinical fellow at South Manchester, has been doing work in humans, looking at human asthma patients and looking at their responses when they present to clinic at different times of the day. And what Hannah's shown, so this is just looking at the percentage of sputum samples that are classed as positive or negative based on their percentage of eosinophils within the sputum. So essentially, patients presenting sort of early morning are a lot more likely to have a positive sputum sample than those presenting later in the afternoon. So, you know, now it's going to be important to try and drill down and understand why this is occurring. Julie's also been doing work in humans, shown there's a similar time of day response in rheumatoid arthritis patients, so that patients coming to clinic at different times of the day will show different different levels of inflammatory cytokines, for example. And the current work is trying to identify the mechanisms by which the biological clocks regulate this loss of tolerance. So using mouse models and then trying to translate that into to patients. And then, hopefully, I've already started to put across that, tolerance is important in very many different organs. In Manchester, we've got a great strength in many of these areas. I've already talked about rheumatoid and the myopathies. Paul Brenchley is also doing some great work looking at inflammatory kidney disease. And as John's already pointed out, we have a great strength in understanding the immunological mechanisms regulating tolerance at barrier sites. So just to highlight a couple of posters that have been presented here. So we have great strength in the intestine. So Felipe Melo Gonzalez from Matt Hepworth's lab, who's deputy for the immune tolerance theme. Felipe is presenting a poster on how innate lymphoid cells regulate antibody production in the intestine and the molecular mechanisms by which this occurs. And Nicholas Scott from Lizzie Mann's lab is presenting a poster on important ways in which antibiotics can disrupt tolerance in the intestine via changes in intestinal macrophages. And Richard Grensis will be talking about this a little bit more detail as well later as part of the commensals branch, again sort of highlighting the crossover and synergy between the different branches. But in the final few slides, what I'll do is just talk a little bit about a study that we're just about to publish, and this is drilling down a little bit more into important tolerogenic mechanisms that occur at different sites. And we have had a particular interest in a cytokine called transforming growth factor beta, which is a really, really important tolerogenic cytokine, but can do lots more than that as well. So just to give you a very brief introduction to this molecule, transforming growth factor beta, it does lots and lots of things. It can regulate many, many different cell processes, and different cells can respond in different ways. Important for Lydia Becker, for the immunomatrix theme, and Judy will introduce this a little bit later as well, is that it's a very important profibrotic molecule. But another very crucial mechanism is that it has a very important tolerogenic function. So this was first shown in mouse models, where if you knock it out, the mouse die because they lose tolerance of their T cells, which start to attack many organs of the body. But this is a very unusual cytokine in that every cell that makes it as an inactive complex. So this is just a schematic of what it looks like as part of the same gene product. What is encoded is this red thing, latency-associated peptide, which is cleaved from the active cytokine but remains non-covalently associated and wraps around it. So this is secreted all over the body, but it must be activated to have biological function. So work that we've been doing for a number of years is to try and understand how, when and where is this important cytokine activated to promote immune tolerance. And I'll just leave you with a couple of slides just highlighting a recent study where we've identified a key pathway that regulates immune tolerance in humans. So essentially what we've found is that in humans, and interestingly not in mouse models, classic monocytes, these important immune cells that are found in the blood that can infiltrate tissues during inflammation and during homeostasis to develop into macrophages. They express a particular molecule called integrin alpha V beta 8, which is a key activator of TGF-beta. We've shown that this expression causes these cells to activate high levels of TGF-beta, and this seems to be an important autocrine tolerogenic signal, because if you block this pathway, what happens is when these cells get stimulated, if you block the pathway, then they make lots more TNF. So this seems to be an important break on the ability of these cells to make TNF alpha when they're stimulated. We've also looked in important barrier sites, so we're interested in the intestine, and what we found is that monocytes within the intestine also express high levels of these TGF-beta activating molecules. And the cells in which these monocytes can differentiate to, so tissue macrophages, also express very high levels of this important tolerogenic pathway. But then what we found is if we take these intestinal samples from patients with inflammatory bowel disease, during severe inflammation, what happens is somehow, and we don't understand why yet, this tolerogenic pathway is very much downregulated. So, and what you also see is you see a shift in the balance of the tissue macrophages to monocytes. So it appears that the monocytes are coming in, downregulating this tolerogenic pathway and promoting the inflamed environment. So just the final summary slide. So immune tolerance is, as I've said, this very broad theme. We have great strengths in the study of the genetic predisposition and the mechanisms promoting immune tolerance. I've given a few examples of how we're taking the genetics and then trying to then relate this to the mechanisms. I think as part of the branch we can strive to improve this and do more studies in this area. But then also, hopefully, I'll put across how broad this topic is and how crosstalk and synergy with all the other branches will be very important, and will highlight new areas of research and hopefully identify new ways in which immune tolerance contributes to a variety of different disorders. So with that, I'd just like to stop and thank you for your attention.